If you're a biotech founder, investor, or pharma innovator trying to accelerate drug screening beyond outdated models, here's the challenge. Traditional tumor models are slow, costly, and unreliable. Ghazaleh, founder and CEO of CanChip GmbH, has built a microfluid tumor-on-a-chip platform from the Potsdam Science Park ecosystem, backed by national startup awards and a mission born from a personal oncology journey. Today, we'll break down how her tumor-on-a-chip innovation can help you bring personalized cancer therapy, animal-free drug development, and predictive tumor microenvironment models into real-world impact so you can leap ahead in the competitive biotech space.
Welcome to Startuprad.io, your podcast and YouTube blog covering the German startup scene with news, interviews, and live events.
Welcome to Startuprad.io. Our guest today is Ghazaleh Madani, co-founder and CEO of CanChip, a Potsdam-based biotech startup revolutionizing cancer research with its tumor-on-a-chip microfluid platform. Actually, you'll explain to us what that means soon, right? With a master's in biochemistry and molecular biology from the University of Potsdam and a bachelor's in medical laboratory science from Isfahan University, Ghazaleh brings rigorous science, entrepreneurial vision, and personal motivation. Her mother's cancer journey led directly to her founding mission. Since 2023, CanChip has developed human cell co-cultured 3D microfluid tumor models, achieved major awards, including Newcomer of the Year at the German Startup Awards 2025. That's why you are here. Congratulations. And is positioned at the cutting-edge intersection of personalized medicine and non-animal high predictive clinical testing. Today, she joins us to unpack how tumor-on-a-chip, microfluids, angiogenesis modeling, and biosensor integration combine to create the next frontier in oncology drug development, and what that means for founders, investors, and pharma partners alike. Ghazaleh, welcome to the show. And I think you really explain a lot here. I do have an audience who listens from time to time to biotech content, but I have to admit it's quite unique. What are you doing? Can you take us along your journey? You started in medical laboratory science in Iran, moved to masters in biochemistry and molecular biology in Germany, and then founded CanChip in 2023. I bet when you started your bachelor, you never imagined that. Could you walk us through this once upon a time moment when you realized that tumor-on-a-chip was the entrepreneur path you felt compelled to take? And can you please explain what a tumor-on-a-chip is?
Yes, of course. First of all, thank you very much for having me here. That's an honor to be involved in such wonderful podcasts. And when I go back and think about my bachelor, I could never imagine that I am here in the position that I am today. And the moment once upon a time for me was actually during my master degree with the idea of CanChip, that we were collecting data from like 2D models, and then we were doing animal trials, and some of them were working really good. And then I was realizing that they are clinically really not relevant. We were relying on models that they do not behave like real human tumors. And that was disconnecting me from the point that tumor on a chip can stop being just in a dream or being in academia and become a mission for me. What is tumor-on-a-chip?
And since we're not doctors, can you tell us why this model behavior that differs from the real human being, why this is a big problem?
Yeah, when we are having like the preclinical data for the pharma companies and biotech companies, they normally have 2D models. What is a 2D model? Is like a flat cell culture, the cells are glowing and they are not acting as they can act in human body because we don't have a flat organ. They are all like 3D, 5D missions. And the other thing is that they are testing in them on animals, and animals cannot behave like human. And they are the reasons that so many of the drug developments are failed in clinical phases, and that is very expensive to develop a drug, that is time consuming to develop a drug. And what these tumor-on-chip models do is that they are 3D. They have the microenvironment of the tumor, so that is really a mini tumor on a biological platform that can be used for testing the medicaments that they are in the development. That in that area, we do not need animal trials anymore so much. So we are reducing the animal trials, and we are also having more reliable results at the end of the story.
Mm-hmm. In the everyday status quo of cancer drug development, which is a very, very difficult field, because we don't know a lot of what actually causes cancer. So that's always a question, how to treat it. We still rely heavily on animal models and, like you said, the 2D cultures. How does your system, the tumor-on-chip, challenge that status quo? And what is the biggest limitation you saw that led you to build this platform?
The way that we challenge the status quo per day is by creating the microenvironment that actually behaves like human tissue, with perfusion, with co-culture, with real-time function readouts. And that means that we have a dynamic platform, like the blood is going through the organs, the medium for the cells is going through them, and the readouts that we have, they are the readouts that you need to develop a drug. And it's just not a smaller model, it's a human model. And the limitation that we saw to lead us through that is, as I said, the results that we get sometimes are a fantasy in 2D model. And we are like, wow, we just developed a new drug. And when it goes to the clinical trial with human, that is not working. And that is the huge limitation, because we are talking about millions and billions of euros that yearly is being spent on drugs. And we are talking about 14 years that one drug is taking from the beginning of the drug development to when it's ready to be released to the market.
Mm-hmm. Mm-hmm. I see. And that's basically where your tumor-on-a-chip really gets started. So, and actually, that it was all triggered, as we already said in the intro, by your personal experience, a personal catalyst. Your mother was diagnosed with breast cancer, and that inspired your mission. How did that emotional driver shape your scientific focus and your founding strategy for CanChips?
I mean, the cancer was for me always, when I was also in my bachelor degree, was for me always a very big question mark. But I realized that when my mom was diagnosed with breast cancer, the science was one of a sudden for me personal. And I understood that every delay that we have in preclinical phase of the drug development translates to real suffering, real world suffering. That means later drugs, later personalized medicine. And it sharpened my focus that accelerating a realistic cancer model isn't a scientific ambition. That is responsibility that someone like me has when they have idea and they have passion to bring it into near world. I mean, that's like being so realistic at the same time being also so emotional, because I can say that each of us had someone in the family that was fighting for cancer or in the friend crisis, and know what a story is that. And when we are one step more forward through personalized medicine, then that is personal for all of us.
And due to this catalyst, you chose to integrate the human cell, what you call co-cultures, perfusion microfluidics, and multi-omics readouts. You gotta explain what that is. Into chip design, what were the major engineering biological pivots you encountered by developing the tumor microenvironment model? So, meaning the setting up a model that the tumor really behaves like a tumor in a human body.
Exactly. I mean, the point is that when we are talking about these microfluidic systems, you can imagine a resin base that we are working with, or some other companies like a microplastic and so on. And we had to think about it that the chip cannot behave like a plastic or a resin. That should behave like a tissue. That should behave like a human. And making that microenvironment to be in that area that was balancing like biology and engineering. And we are really biologists and biochemists by heart and by practice. And that was the irritation that we had to mix these two together. And that was the moment that I was like, okay, that is what can be really good, because we found a way to have zero drug absorption in our platform, to have a transparent platform, and to have this engineering to come in a real world data, that we don't talk about just the chip, we talk about like real tissues.
We're also going to record a founder's world, of course. And you once told in an interview that we need a quick shift towards personalized cancer medicine and we must leave our comfort zone. You'll there share moments when you personally pushed yourself beyond that comfort zone and what that meant for CanChip's growth. Until finally, after prototyping and validation, CanChip achieved proof of concept for what you call tumor-on-a-chip models and secured its first awards and partnerships. What were the breakthrough milestones you hit, and how did you validate your platform to pharma and biotech collaborators?
After this time of the prototyping and so on, we were really in a point that we were ready to go. And I have to say that our breakthrough was consistency. There are so many challenges, daily challenges that you have in biotech. And specifically when you are managing to have a unique and innovative platform or a service, when we reproduce the same drug response curves in independent runs, we could show the biotech and pharma companies that this platform is ready. You can trust it because the data that we have, they are clinically relevant, because the data that we have shows that you can reduce the animal trials and you can have better results faster. And that was the moment that we were coming to talks with biotech and pharma companies to understand their need, to also push them a bit out of the comfort zone and tell them, let's make it a bit bigger together. And that was a journey, but that was going good.
Mm-hmm. Mm-hmm. I see. From your platform's perspective, what are the tactical frameworks you deploy when designing those 3D tumor-on-a-chip models? For example, tumor and co-cultures, perfusion settings, and real-time biosensing. How does this differ from the standard approach?
The way that we do it is normally with mixing some points together. For example, that is the architecture part, as you said. We have all the microenvironment. We just don't have the tumor cells because, that doesn't work as it should. We have the tumor cells. We have the endothelial cells. We have the immune cells that it should be included. Then we have the perfusion model, that we have the translation of the flow that we have like in human body with the blood. We translate it into the tumor-on-chip model, and we have it on for the drug development because the absorbance will be different, the effect and side effects that it has, it will be different. And then was the validation and the readout. The difference that is with the standard assays is that is more complex, so that there are different questions that can be answered with using that platform for each type of tumor.
For our audience, because I believe you and me, we had way more touch points to cancer than we would ever like to have, you, hopefully mostly in professional manner. For everybody who's not with a medical background in oncology, how many types of tumors out there? I do believe there's so many that even oncologists encounter from time to time a type of tumor they've never seen before.
That's true. They are really, for each type of tumor, there are so many type of subtumors. When someone has, for example, colorectal cancer, the other one also has it. The subtypes can be totally different. That is genetic based, that is based on the mutation that the cancer has. And I say it always, if 100 companies at the same time work with the platform that we have, still each of us has so much to do, because there are really so many areas of cancer that they are non-touched, that they are the cancers that you understand at the end stage and you cannot do something about it. So I really have to look it up exactly how many types of tumor we have, but I can assure you that there are plenty, and so many of them are still so much unknown to us.
It's quite incredible how many different types of tumor one can have, and every subtype even needs to be treated differently. So it's a huge field, and a lot can be gained by having the right therapy. So going a little bit back to CanChips, what are some of the scaling challenges you've faced, both biologically and operationally?
I mean, biologically, I have to say we didn't have so many validation problems, the reproducibility of the system and so on, because my co-founder also had so much experience in the cancer research and microfluidics. We were really going a bit more faster. But the biological challenge that not us, but so many of the companies has is that the variability of the primary human cells, that when you are working and saying, I am developing a pancreas cancer on chip, you are developing one of the pancreas cancer models or sub-models that is even possible to study on, and there are many, many different more to go. And that was also the maintaining the barrier integrity across the chips, that you want to have so many different cells on the chip, each of them grow differently, so you have to control some of them, you have to let them grow freely, and they are the biological aspect of that. And operationally, of course, that was the early regulatory talks, because we know that FDA also accepts these non-animal models, but there is not golden pathway that you can say, if I go this direction, everything's accepted. The good point is that because we are in the preclinical phase, we do not have so many regulatory aspects that we have to go through. But still, if you want to convince biotech and pharma company to do less animal trial and come to these non-animal models, they need to know which direction they are going. We are active in this area with FDA, with EMEA, to really try to make it a pathway for all of us. But I have to say, operationally, these regulations and convincing is a challenge.
And when you talking about FDA and so on, that are actually the regulatory bodies for Europe and the US.
Yes, exactly. Whenever a drug is coming to a market, they should definitely have the FDA approval to be allowed. That means that they pass the preclinical test, clinical test, and the drug is safe to be in the market for patients.
I was wondering for our audience, if you're working on oncology R&D or biotech investments and you're hearing this, what would you ask Ghazaleh right now about tumor-on-a-chip models that can impact your own project? Guys, we'll be right back after a short break, dive into strategic decision-making and customer partner wins that really propelled CanChips forward. You mentioned you're working with patient-derived tumor cells and integrating biosensors for real-time drug response monitoring, which is actually pretty cool that one can do that. Can you walk us through a case study or partner collaboration where this played out?
Of course, we have a very, very good collaborator that really trusted us from the very beginning, that we use the patient-derived colorectal tumor cells on our chip with continuous perfusion. And we were monitoring the drug response in the real time through the sensors that we are integrating, and also that time through the gene expression and so on. And we could discuss this manners, how the cells are reacting with that partner. Instead of waiting days for the results, we could have in 48 hours, 72 hours, the primary results that we wanted, and that is gonna change the decision making, that is gonna change the personalized medicine. What we want, I am gonna say it so often in every interview, till we see a change, we want more collaboration from hospitals in Germany to go through personalized medicine. That is not a one-man show. We have to work all together. We receive samples from Sweden, from Romania, from all the Europe. But when it comes to Germany, they are like this protection of the data, even though we don't need the data. And that is the place that we really can develop the personalized medicine. So we have to keep it together.
Mm-hmm. You were talking about a 48-hour time frame here. With current methodology, not your tool, how long does it take?
In general, when we are integrating models, that is a bit dependent. When we have a patient drive and they are really well biopsied and we see that there are cancerous cells inside, they can directly go to the chips. And in 42 hours to 72 hours, we have the base that we wanted to have it. But sometimes we may need longer because some cells are not growing in the speed that we want them to grow. But I always say we will have a 2-week time since the patient is operated and the tumor is out to the time that they are ready and recovered for the therapy. And in these 2 weeks, we can answer so many questions that can help them for personalized medicine.
We're here on a podcast where more than 90%, according to our audience survey at least, listen for professional reasons. So let's dive a little bit into your strategies. What growth methodologies did you adapt in your early startup phase to integrate science, engineering, and startup execution? Because many know the saying in startups, move fast and break things, but that is not really working in cancer treatment. So what can a biotech founder learn from that? What did you learn?
I mean, I have to say a very, very early point is that the startup founder should be really brave enough to be loud about what they are doing. Because if you are developing the coolest product, but no one knows you, there is no worth for that product. And I learned it in a hard way because our investor was like, you have to go out and tell the story what you are developing. So being loud is the first thing that I always say to the founders, as then is to be fast. You know, you cannot be perfect. You have to get out the first prototype, let the end users try it, even in a pilot study. Then you understand what is the issues, what is the challenges, and you can make it. And of course, the co-development with users. You know, I always say that it's not a one-man show. You need collaborators, you need people in academia, in other industry to help you understand what is the need of the market even. And of course, a very good business developer to get in touch with potential customer, because you can develop something for them and that is not what they wanted. But if you can ask them, what is your need? What do you want? Then the product that you're going to develop is the thing that they're going to be interested at the end of the story.
Mm-hmm. Mm-hmm. And now dive a little bit into your strategic decisions. For example, for you, it's a strategy decisions what cancer types to target first. I think high on your wish list was, of course, breast cancer. I'm very sure about this. Also setting your RP strategy, prioritize co-cultures and so on. What decisions did influence your roadmap from 2023 to, let's say, next year, 2026 and beyond?
For us was the choosing of the cancer type and targeting was really dependent on the, unfortunately, I have to name it, trend. That's a very sad story of the sicknesses and cancer types that they are getting more and more, and they need really more studies. That is colorectal cancer that is coming to younger generation, pancreas cancer, and the cancers that they are mostly in the pipeline for treatment for pharma and biotech companies. And that was for us the point that we said, okay, we will start from that point. The IP strategy for us, I mean, we already filed the first patent in 2024 because that is also a thing that we are doing is novel. So why shouldn't we protect it? And the plan or roadmap that we have for 2023 to 2026 for us was influenced by really making something big that different companies, with, if they are small or if they are big, if they are universities, that they can benefit from that. And that the goal is to really get away from animal trials, and we are also influenced by that mindset as well.
Mm-hmm, mm-hmm. What customers or partner wins stand out for you, like university labs, CROs, pharma, and how do you quantify the value your tumor-on-a-chip platform delivers to those collaborations?
The partners that we have till now really valued our models because they are less variable and they are more predictable. And the reason behind that is that we have customized models. So we ask them, what is your need? And then we will design the platform based on their need. And that is what a bit our standards. And one group like reduce the time that they had to put on the animal trials by around 30%. The other one gained functional readout that they couldn't do. And they are the things that we can quantify the value of the tumor-on-a-chip with the feedbacks that they gave us. They said, I could understand a problem in my drug development that I am struggling with since years with animal models, and I couldn't see where the drug is going. So they are the ways that we understand that we are in the right pathway.
Mm-hmm. When I was putting this interview together, I had to admit I have not the slightest idea of what people you need for such a cutting edge technology. So I'm considering funding and team building. What are key criteria and talent profiles you're prioritizing now, especially given that specialized nature of microfluidics, biotech, and oncology, I'm very sure you won't find a lot of bachelors and masters, people with a specialized track for this specialties, right?
Yes, of course. I mean, that is an area that is really needed, like education in that area and also lab work in that area. So when we are also planning our expansion, and our team is going to definitely be people that they have already worked with tissue models. So if they don't have experience in microfluidic, that is not an issue. We can bring it for them. And the like researchers that they are in the translational model. How can we translate these in vitro models, the models that they are happening outside of the human body, to the clinical models, that what is happening inside human body without directly testing it. So these are the areas that we mostly look for our expansion, and of course business developers, because as I said, if you are developing the coolest product, no one out there knows what you are doing, so you cannot really bring it to the market.
Understood. Let's go a little bit in the future outlook. How do you see the tumor-on-a-chip and organ-on-a-chip markets evolving by something like 2030? Where does CanChip position itself in that landscape?
I have to say that the organ-on-chip will mature itself from innovation to infrastructure. That is where the regulatory affairs are also going. So I think and I hope that by 2030, it will be a standard in early drug discovery, because since 2024, so many things happened from the day one that the FDA said, I am going to accept these models, and we position CanChip as a leader in realistic tumor-on-chip models, ready for precise oncology and ready to have customized solution for pharma and biotech companies. By 2030, we are a leader.
I'm always trying to tease out a little bit of contrarian view here. Many believe larger animal-based preclinical models will always dominate due to regulatory inertia. Why do you think that belief is broken, and what evidence do you have to challenge it?
I mean, the regulatory interior is always real, but the science is stronger. And we don't have to forget that the science is moving very fast lately. Animal models simply do not replicate human body anymore. They do not replicate patient response enough. The field is moving toward human relevant system, and regulators are also acknowledging that. And when we think about combining the AI data that we can have around patients, our models and genetic models all together, then none of the animal models can really compete with that. That is my point of view on this case.
For our listeners tuning in, I would be curious, what is one immediate question you would challenge yourself to act on this week if you would be in Ghazaleh's seat? Be bold. Leave a comment here. Let's talk a little bit about advice for founders. If you're listening to scientist, entrepreneur, or deep tech founder, what are 3 mistakes they should avoid and 3 practices you should adopt to navigate translation from a lab to commercialization? Whew, hard word.
I mean, the 3 points that I would really would tell them to avoid is to wait for the perfect data. There is no perfect data. To not overbuild the tech before talking to users. Sometimes there are some platforms that they are so complicated that they cannot even work with it, and underestimate the complexity of the manufacturing and the steps that you have to take. That is always, even if it's about time or if it's about cost, make it a bit more than you think it's enough. And points to adopt is early pilots, to bring your product or prototype out to see what is going on. The IP strategy, of course, to think about it and really secure it, that is going to save you, and prioritize the things that are important for the company without thinking about what is going to happen if and if this prioritization is really going to make a huge difference.
We getting very close to the end of our interview, and I would be interested, if CanChip overachieves, what is the one breakthrough in personalized oncology we look back on in 2030 and say, that came from CanChips?
Yes, that would be the drug response prediction that matches the patient. That is what I always say. That is matching the patient, that is not matching the animal, that is not matching a 2D model. That's how personalized oncology become real. That we can say this patient needs this specific drug, they get it, they will get so healthy, and the cancer will never come back again.
That would be great, because a lot of people are still dealing with cancer coming back and coming back and coming back. That's really a tough spot to be in, because it always gets harder to fight back. That was a depressing note. I'm so sorry. We usually close our interviews with 2 standard questions. So, number 1, are you open to talk to investors?
Yes, I learned it also that I should not close any doors, because that is not just about money. That is about the network. That is about the knowledge that the investors also bring. So for a talk, I am always open.
And of course, the usual question, are you looking for talented employees?
I have to say that I have enough CVs that I can hire minimum 10 people by tomorrow, and they are highly educated. And the point is that because we are an English-speaking company, that makes it easier for them to approach to us, because this language barrier is real here. And we are expanding, but I already picked the people that I want to hire.
And we learned a pretty big secret to get good talent here in Germany. If you are an English-speaking company, So we finally teased something really cool out. Ghazaleh, thank you very much. Best of luck for your future mission for CanChips, and congratulations again for being one of the winners of the German Startup Awards 2025.
Thank you very much. Thanks a lot.
That's all, folks. Find more news, streams, events, and interviews at www.startuprad.io. Remember, sharing is caring.